This is a video reviewing the TCA cycle or the Krebs cycle. As a reminder, we're starting our story with glucose, which is a 6-carbon molecule. The bonds of those carbon have stored energy that we need to be able to get out to use for metabolic work. In the process of glycolysis, out in the cytosol, the 10 enzymatic steps of glycolysis were able to strip off 2 ATP and 2 NADH, ripping both energy and electrons off of that glucose molecule. And at the end of glycolysis, we ultimately ended up with 2 pyruvate molecules. The reason there are 2 is that glucose was a 6-carbon molecule, and in the process of glycolysis, we split that into 2 separate 3-carbon molecules. Now, to get the rest of the energy out that still remains within those bonds between the carbon, we're going to have to bring the pyruvate into the mitochondria. It actually has to get transported all the way into the matrix, so past both of our membranes of the mitochondria. We have to get our pyruvate in, and within the mitochondria, pyruvate will be converted to acetyl-CoA. So both of these will have to move their way in and get converted to acetyl-CoA. The enzyme that does this conversion is pyruvate dehydrogenase, an important enzyme that converts pyruvate to acetyl-CoA. It does strip 1 electron off, so we're able to get an NAD converted to an NADH at this step, at this conversion point. The other important thing to note is that this 3-carbon pyruvate is going to turn into a 2-carbon acetyl-CoA. So in the conversion, we are also going to strip off a carbon dioxide. This is the first time we've talked about carbon dioxide. It's the first step in glucose metabolism where we produce one of our final end products, which will be carbon dioxide. So this 2-carbon acetyl-CoA has the capacity to join the TCA cycle within the matrix of the mitochondria. As long as there is an oxaloacetate that is available to bind, oxaloacetate and acetyl-CoA can come together to form citrate. Oxaloacetate, or OAA, is a 4-carbon molecule. So after it combines with acetyl-CoA, citrate is a 6-carbon molecule. Then there are the steps of the TCA cycle, which are a series of enzymatic reactions which will alter the chemical compound, ultimately stripping electrons and ATP off of this 6-carbon citrate. We're not going to memorize the steps of the TCA cycle, but it is important to know that at 3 different places, we take an NAD and load it up with an electron, removing that high energy electron, which will ultimately be used by the electron transport chain to make ADP. There is 1 step where we use the electron carrier FAD and load that up with electrons. So 1 turn of the citric acid cycle produces 3 NADH and 1 FADH2. There are 2 spots within this cycle that produce carbon dioxide. So we strip off 2 carbons, which makes sense because our final end product, oxaloacetate, only has 4 carbons left. And it becomes a 6-carbon molecule when it rejoins with acetyl-CoA. So here are the carbons that got stripped off. In fact, from the original 3 carbons that were pyruvate, we can see 1 stripped off in the conversion to acetyl-CoA, and then 2 more removed in the TCA cycle. With all of these actions of the TCA cycle, we've done a lot to remove potential energy from this molecule, but we haven't yet made any actual ATP. There is one step in here that produces a GTP, which is another high energy molecule within a cell. We can think of this as an ATP because that GTP is used to produce an ATP. So from one turn of the TCA cycle, we get one single molecule of ATP. So this is a necessary step to make the electron carriers that fuel the electron transport chain. But in and of itself, this pathway doesn't produce a lot of ATP. It's mainly a mechanism to feed the electron transport chain.